In order to understand cell biology, you really have to have a good understanding of cell division and how an organism needs to either A, make copies of itself, or B, make unique gametes, or egg and sperm, in order to reproduce effectively. Or in the third scenario, they just may have to regenerate some new cells due to an injury or damage to a portion of tissue, or perhaps there's a developmental change to an organism's body that's been triggered. So, regardless of the scenario, cell division is a really important part of understanding how organisms grow, develop, repair, maintain, and also reproduce. So, in this slide presentation, I'm going to cover all the important components of cell growth, cell division, and how organisms achieve these tasks. So, to start, cell division in prokaryotes and eukaryotes do differ. A couple of different things that occur in eukaryotic cell division that are much more complex and has a more elaborate process. So, you think about the cell structures of a prokaryotic organism, they are very simple, they're much smaller, they don't have all the complex structures, so it is definitely a different process. So, in prokaryotes, the cell division process is known as binary fission. In this process, the cell basically grows and increases in size to its maximum capacity, and then it's programmed to divide under the process of just splitting into two daughter cells that are genetically identical to the mother cell. So there isn't any genetic diversity created in the process, but it does have its advantages. It doesn't require a second mate. It also doesn't require much time. It could be very, very quick. Some organisms can complete binary fission in just the span of 6 hours, while some organisms may take up to a full 24-hour day period. But overall, it's a very fast process. Cell division in eukaryotes, obviously, they have much more for cell structure. They're much more complex, so it does take a few more steps, and it is much more elaborate and definitive in its steps, and it has sequence and order. And there's a reason for that we'll cover. So in prokaryotic organisms, binary fission is the basic premise of how they divide. You basically have a starting mother cell that is going to grow to its maximum capacity, and then it's going to be programmed to duplicate its DNA. And if you can see it here in this picture, I'll try to zoom in a little bit. You can see that the prokaryotic cell has copied its DNA in a single step, and then in the second step, the cell begins to divide. You'll see that two cells are going to be the product. You could see that part of the DNA has gone to one side and then the other part of the DNA to the other. And upon division, you actually have two identical cells. Now, they're not going to be haploid, they are going to be diploid in this case. So that's something important to identify, that you understand the difference between haploid and diploid, which I'll get to more later. So this is the basic premise of what takes place during prokaryotic cell division. Each new daughter cell is going to receive its genetic components, a portion of the cytoplasm, a portion of the membrane, to be its own living cell. So in eukaryotes, the process is a little bit different. You actually have a step-by-step process that is much more elaborate and distinguished from that of binary fission in prokaryotic cells. So it starts with interphase and then goes into the M phase, which is mitosis, and then cytokinesis. So because it has so many steps and because it has many subphases, it does take much more time. But there are some unicellular eukaryotic organisms that do this, as well as many multicelled organisms that have large amounts of tissue or organs that require maintenance and repair, as well as having development and growth as well. So the first step in the cell cycle is interphase, and you can kind of see in this image that interphase encompasses G1, S phase, and G2 phase, and something different happens at each stage. So during one full cell cycle, each cell is going to have to undergo each of these steps. And you could see that, how long the cell spends its time in is kind of annotated by the pie graph there. So if you look at the G1 phase, where there's primarily cell growth, it's a pretty nice chunk of time, as well as S phase, when the DNA is copied, that is time consuming, as well as G2 phase, where there's additional growth and preparation for cell division. There's some proofreading that goes on from the DNA replication, as well as enzyme production for mitosis. So if you count G1, S, and G2, you'll see that it takes a very large portion, almost 90% of the time that the cell is going through development and growth. So it's a very pronounced amount of time, very very noticeable. So that's the longest stage of the cell cycle. So interphase is broken down into three subphases. You have G1, uh, often referred to as gap 1, and in this particular subphase, the cell is going to increase in size. It's going to add cytoplasm, cell membrane, increase its organelle production, pretty much getting the cell, all of its contents to be split into two. And then after that's completed, there's a checkpoint, and then there's S phase when the cell's DNA is duplicated. So now it has double the DNA that it had previously. Now that makes sense because you're going to be making two cells, so you need double the DNA. During G2, this is called gap 2. The DNA that was duplicated during the S phase is going to be proofread and checked for genetic mutations or errors, and they'll be corrected at that time. Also, if there's some additional secondary growth that needs to be made, there'll be some increased production of cytoplasm, maybe some organelles, and also the enzymes needed in mitosis. And then the G0 phase is sometimes overlooked. Some people don't mention it, but it is a stage that's kind of like a holding phase that the cell will sit there and wait until it is triggered to begin the process of mitosis. So here's just kind of a drawing here of a cell. You could see that this cell is an interphase. You could see the cell membrane on the outside perimeter. You could see the nucleus is present, the darkened nucleolus in the center, and then you could see centrosomes that are on the outside of the nucleus. Those are going to be important during mitosis, so they are produced in interphase. Those are kind of the noticeable things you'll see in interphase. Now, the M phase or mitosis is pretty much where the nucleus is going to go through some different developments. Its appearance is going to change. Chromosomes are going to go through some different things, and then the nucleus will actually divide. And this is when you actually get cytokinesis occur, and you can have the two daughter cells are produced at this time. So the four subphases of mitosis are prophase, which is first, then metaphase, then anaphase, and finally telophase. So 4 phases of mitosis. So here are what the 4 phases look like. On the left you have that's early prophase. You can see that the nuclear membrane is still present, but you can see that there have been chromosomes that have condensed and formed, so those wouldn't be present during interphase, but they are present in prophase. In metaphase, the chromosomes have lined up along the center of the cell, and that's very pronounced and noticeable. You can see the spindle fibers in the centrioles have taken a very specific location and where they're going to be anchoring those chromosomes. And then during anaphase, you can see that the chromatids have been separated and they're being pulled apart, and they will be isolating to opposite sides of the cell. Oftentimes you may even see in late anaphase that a cleavage furrow forms at opposite sides of the cell, kind of a pinching of the membrane. And then in telophase, the cleavage furrow is very pronounced, and you actually can see the formation of 2 daughter cells is imminent. And there are 2 new daughter nuclei formed as well. So each of the stages has a different appearance, and they are quite noticeable, so you can distinguish them. So prophase, the first stage of mitosis, it's pretty easy to spot when you see a cell in prophase. Here it is, and you could see that early in prophase the nuclear envelope is still present, chromosomes have condensed, so that's kind of the noticeable thing about prophase. Pause the video if you'd like to see the individual steps there. You can continue to read here, just hit pause on the video. So here's what the prophase step looks like in color. You can see the chromosomes have formed, they have condensed. You see the spindle fibers as well as the centrioles are moving. Here's what prophase looks like in a plant cell. Cartoon drawing. Now metaphase is the second phase of mitosis, and you can see that the spindle fibers have positioned themselves in a very strategic way, and the centrioles have been positioned at opposite ends of the cells. In the process, they're going to align the chromosomes along the metaphase plate, or the equator of the cell, and you can distinctively see that they are lined up in the center. So here's another color drawing of what that looks like. And this is what it looks like in a stained sample of a plant cell. Now the third stage of mitosis is anaphase, What happens during anaphase is pretty much this is where you get the division and the separation of the chromatids that are attached to each other. You could see in this cartoon drawing that the chromosomes have been pulled apart and separated, and of course this happens because each new daughter cell is going to need its complement of DNA. So here's a color animation or drawing of what that looks like. You can see that the chromosomes have been separated and they have moved and migrated to opposite ends of the cell. Here's what it looks like in a stained sample of a plant cell. And the last phase of mitosis is telophase. And in telophase, you can see that the chromosomes have already been pulled apart in anaphase. The nucleolus reappears on each side onto each cell. The cleavage furrow is very pronounced. All that's left to do really is the pinching of the membrane so you can get the separation of the two new daughter cells. And that's what it looks like in the case of a plant cell. Now what's important to notice, if you see the structure between the two nuclei, you can see that there is a very visible divisional line, and that structure is called the cell plate. The plant cell wall is very, very rigid. It's made of cellulose and it's very tough. And without the formation of the cell plate, cytokinesis cannot proceed and you can't get a pinching and separation of the two daughter cells. So in a plant cell, in order for telophase and cytokinesis to complete, the plant cell plate is going to be critical in forming, so that way they can actually separate. So there's a good visual of what that looks like. You can see the two daughter nuclei completely separated, the pinching, the cleavage furrow, it's all there. And then finally you have cytokinesis. This is where you actually get a division of the cytoplasm, and it's very noticeable. You can definitely see that the two daughter cells are present. There's the cell plate in the center between the two cells that are forming between the original plant cell. So there it is in a step-by-step sequence. That initial cell on the left-hand side, that would be during early prophase. The nuclear envelope is still present, but the chromosomes have formed. And then you could see in metaphase, the chromosomes do line up. And then in anaphase, they start to split apart. Chromosomes are going to separate ends of the poles. And there is a cleavage furrow form at the top and the bottom there. And then there's late anaphase in the bottom right-hand corner. And then you actually get telophase in the bottom left-hand corner, where you have two new daughter nuclei. The cells have completely separated from each other. All that's left is the cytokinetic event, where the cytoplasm divides and you have two new daughter cells. So here are just some images. A lot of you have already seen this with the plant cell samples we looked at in the microscopes. You can see the shape of the plant cells is pronounced, but you can also see that this is probably an onion root cell sample. You can see that each cell is kind of in a different stage of the cell cycle. So some are in interphase, some are in prophase, metaphase, anaphase, telophase, and even cytokinesis. So, but what you'll notice is that most of them are in interphase, and that makes sense because cells spend the most time during interphase. So the last section of this video is going to be on meiosis, which is gamete formation. This is a little bit different than mitosis. It does have 2 rounds of division as opposed to 1. Gametes are formed, not body or somatic cells. So in the case of mitosis, we're talking about cells that are somatic or body cells. They are diploid, and they start diploid and they end diploid. In the case of meiosis, these cells start diploid and end up as haploid. So there's going to be some distinguishable characteristics of each of these stages that I'm going to highlight. So in meiosis, you basically are going to see that the chromosome number that a cell starts with is going to undergo 2 reduction divisions. So in the process, the chromosome number is going to reduce twice, and then the divisions are going to happen twice. So examples of cells that undergo this process are gametes, and the gametes would be egg and sperm. So we're going to continue to highlight that throughout this video. So at the start of meiosis, the cells still undergo interphase. They're going to go through G1, S, and G2. So that's identical to mitosis. But there are 2 rounds of division that follow. So there's going to be meiosis 1, which has prophase 1, metaphase 1, anaphase 1, and telophase 1. And then a cytokinesis event, where you get 2 temporary cells. And then in meiosis 2, those 2 cells undergo another round of division of steps. So prophase 2, metaphase 2, anaphase 2, and telophase 2. And then a second cytokinetic event where we actually get 4 haploid gametes. So here is meiosis 1. Starts with interphase, and you start to see cell growth and all the normal steps of mitosis that we saw, or the cell cycle, excuse me. So you get S, where the duplication of the DNA after G1, and then you get the G2, and then prophase can start. Now, something important that I want to highlight is during prophase 1, you get an event called crossing over, where homologous chromosomes line up and become very, very closely associated. And I'm not sure if you can see it very well in this, but the chromosomes do orient themselves next to each other, and they exchange bits and pieces of DNA. And in the process, it creates recombinant DNA. And that recombinant DNA is the first source of genetic variation in sexually reproducing organisms. So that crossing over results in recombinant DNA, as the tetrads have closely associated each other on top of each other and exchanged the bits and pieces. And then you could see in metaphase 1, the chromosomes are paired up and they are aligned, just like we saw in mitosis. But what's kind of interesting in anaphase 1, during meiosis 1, the chromosomes are not separated from each other and pulled apart. They're actually just going to be held together and still kept together as paired chromosomes, but they aren't pulled apart from each other. So what's kind of interesting in this scenario is the diploid number is still going to be retained and conserved. So you are going to see, in this case, that the chromosomes are going to opposite poles, but they're not being pulled apart at the centromere. So at the end, in telophase 1, cytokinesis 1, you can see that there are still diploid cells being formed. So the products of meiosis 1 are 2 diploid sex cells. Now, they're intermediates. They're not finished. They're not done in division. But what happened in anaphase 1 is different than what we're going to see in anaphase 2. So that's important to note. So this goes over everything that happens during prophase 1. You can pause the video if you'd like to see. So this is the result of the crossing over event. You can see that the chromosomes are lining up and exchanging little bits and pieces of DNA, and that's where we get the genetic variation that occurs during prophase 1. Now, crossing over does not occur in any other steps in cell division in meiosis. It's only during prophase 1. Alright, so metaphase 1 is the next stage after prophase 1. You can pause the video to see what takes place in that stage. Anaphase 1 proceeds after metaphase 1. Pause the video. There's a picture of anaphase 1. And then telophase 1 proceeds after anaphase 1. Now, anaphase 1 and telophase 1 have completed. I now have 2 temporary sex cells that still need to continue to go through the meiotic process. And the next stage that's going to occur, there is not an interphase 2, so it's just going to start with prophase 2. So you've got 2 cells, you can see them top and bottom on the left-hand side, 2 cells that are going to undergo prophase 2. There is no crossing over event, so we're not going to see additional genetic interaction. We're not going to get any more variations of genes at that point. There's metaphase 2 in the second step. You can see that the chromosome pairs are lined up. And then during anaphase 2, if you could take a look there, during anaphase 2, the chromatids actually do separate in this stage, which is different than in anaphase 1, where the pairs only separated to opposite poles. So here they're separated and pulled apart from each other, and you could see that at the end of telophase 2 and cytokinesis 2, I will have 4 genetically unique haploid gametes. So much different than what we saw in mitosis. Meiosis involves 2 divisions, 2 reductions, and a different set of events that occur. Now, the names of the steps are identical, so that doesn't change, but what happens in a couple of them is very unique. So these slides highlight what takes place in each of the meiosis 2 steps. Tells you the products. So here's a good picture of meiosis 2. You have prophase 2, metaphase 2, anaphase 2, and then telophase 2, and then what you have at the end of meiosis 2 is 4 haploid gametes. So meiosis is the formation of gametes. This is how gametes are produced. So you may start with an oocyte, which in the case would become an egg and 3 polar bodies, or it could be spermatocyte, which would, go through meiosis and become 4 haploid sperm. So here it talks a little bit about that process of spermatogenesis. So that's pretty much the overview of spermatogenesis, where you have an original male sex cell that turns into 4 haploid sperm. And in the case of the ovaries with oogenesis, same scenario. You go through 2 rounds of division and meiosis, but what you have at the end of meiosis 2 is 1 haploid egg cell and then 3 haploid polar bodies. The polar bodies are non-functional and they are not eggs. There is only 1 egg produced in oogenesis. So in asexual reproduction, something to keep in mind is all of meiosis is for sexual reproduction. In the case of asexual reproduction, you don't have 2 parent cells involved, so there's no reason for the haploid number to be achieved at the end of meiosis. So in this case, you only have 1 mother cell that's going to undergo any type of reproduction. So most unicellular organisms go through this process. They utilize either binary fission or mitosis to achieve this. So they go from diploids to diploids, which is different than meiosis. Now, sexual reproduction, on the other hand, utilizes the meiosis process, where you have sperm and egg gamete production, and upon fertilization, you have the haploid gametes joining together to make a diploid zygote. So that diploid zygote has a full complement of DNA and chromosomes. So a common question that students ask is, why do sex cells undergo 2 reduction divisions? And the answer is when they restore during fertilization, they go from haploid gametes to diploid zygote. So in order to have the appropriate chromosome number and have the correct chromosome number, they have to reduce to haploids. Then when they recombine, they restore the diploid number to 2n. So here's an overview of mitosis and meiosis. You can see on the left, you have the basic premise of cell division. So eukaryotic organisms undergo meiosis for sex cell production, but for their body cells or somatic cells, they'll use mitosis. Also, prokaryotic organisms will either use binary fission or mitosis for their growth and division. Here are just some pictures of some slides that are all in different phases of mitosis there on the left-hand side and the right-hand side. Here's a highlight of the cell cycle, starting with G1 in the yellow, then S, then G2, and then the steps of mitosis, and then cytokinesis. A little bit about the structure of a chromosome. If you look at the bottom photo where it says A, you could see that and this is actually a pair of chromosomes attached. So you have the centromere, which is a structure in the center of a pair of chromosomes that keeps them connected and attached to each other. Each of the 4 corners of a tetrad is known as a sister chromatid. So those chromatids are the close associations of genetic material that chromosomes take their form as. So in B, you could see that as a single chromosome, and in A, you see a pair of chromosomes. And chromosomes are made of chromatin. Chromatin is a conglomerate of DNA wrapped in histone proteins, and it's a close association that allows DNA to supercoil and become very condensed, and this way that the cell can fit all of its genetic material into the nucleus. So here's some of the stages of mitosis highlighted again. You can pause the video to take a look at what's going on in each step. There's metaphase, anaphase, telophase, and cytokinesis. Now, in animal cells, it's more to note that there is no cell plate because animal cells don't have cell walls. So that's important to note. So the pinching of the cell membrane is very easy in the separation of the cytoplasm. In plant cells, the cell plate is critical for the formation of the two daughter cells. So there it is highlighted there. It forms a vesicular line, forms the cell plate. All right, so if you would like to test your knowledge and see how you do, so if you could guess what picture A, what stage it is in, that is definitely telophase. You could see that the cytoplasm is starting to divide. You have the two daughter nuclei. B, that cell is actually in prophase. So the chromosomes have condensed. See, the spindle fibers and the centrioles are taking form in their specific locations. Cell C is an anaphase. The reason you would know is that the chromosomes have been separated and pulled apart from each other and they're moving to opposite poles. And cell D is in metaphase. Chromosomes have lined up along the equator in the center along the metaphase plate. All right, so that takes care of all of cell division. So cell growth, cell division, mitosis, the cell cycle, as well as meiosis and gamete formation. Uh, this information is found in your textbook in chapters 10 and section 11, or excuse me, section 4 of chapter 11 in the textbook. So if you want to revisit images and reread the text, I definitely recommend it. If you have any questions, just leave them in the comment section below in preparation for your exam. Thanks for watching, guys, and I hope this helps. Let me know if you have questions in the comment section, and I'll see you guys there.